Okay, so what have we got going on? We don't have an exam next week. We have an exam the following week on Wednesday. So there's a quiz next Friday. As I've been doing, there'll be a Thursday help session on Google Meet. I also am available for Monday, Wednesday help by appointment. Okay, and that's 1 to 5 p.m. And that appointment can be made on Navigate, or it can just be made in class the day of. Just tell me, hey, I want help later on. And the worst thing that would happen is that someone in Gen Chem would have already taken that time. Okay, but so far it hasn't happened. Let's see. Okay, that's what I've got. Does anybody have any questions? Help session Thursday, Google Meet, 8:30. Yeah. All right, then what we're going to do is we're going to start off talking about about buffers, because I missed buffers in the last section. And buffers are important in biological systems. So what buffers do is buffers are mixtures of acids and conjugate bases that help maintain pH at a certain level. So we care about that in lab, and your body cares about that too. Your blood is actually maintained at about pH 7.4, ends up being about 7.6 in your lungs, and ends up being about 7.2 in your tissues. Why would it be more acidic in your tissues? Lactic acid, right. So it's not actually lactic acid, but that's the right thought, right? Metabolism itself makes acid. Okay, if you actually, in second semester, we'll go through this, but if you actually go through glycolysis and you look at the production of lactic acid, it's not actually you'd get it from pyruvate, so you get it from, yeah, pyruvate, but it's deprotonated pyruvate, so you don't actually make any acid to make lactic acid, but you do make lactate. It's just a misconception that's in lots and lots of textbooks. But so yeah, you're making metabolism, just making stuff, and you're moving around and things. That's going to make acid, and so that's why your pH is lower in your tissues than overall in your body, and certainly lower than the lungs. So what I want to do is look at the bicarbonate buffer system here. So we've got carbon dioxide plus water can combine to make carbonic acid. Okay, and then that can deprotonate to make bicarbonate and a proton. All right, so tell me why. So I said pH was higher in your lungs, right? So once again, it's always tricky with pH. Higher means more or less protons. Less protons if we're at a higher pH. So why is the pH higher in your lungs? Can you use this to tell me? What about carbon dioxide? You're expelling carbon dioxide. Exactly, you're breathing carbon dioxide out. And so if I breathe carbon dioxide out, that goes down, right? And which way does the equilibrium shift? It's going to shift left, and it's going to use up the acid to shift left. Does that make sense? Okay? So that's why you see that. And this is one of the ways your body will maintain pH in your blood. You expel more carbon dioxide if your pH gets too low. And you do that by just breathing more. Thank you. Did both of them make them all the way around? You guys are so good. All right. So this is your bicarbonate buffer system. What could cause this not to work? Well, you can't breathe. Yes, that would be a problem, right? Yeah.
Not enough water, like you're dehydrated.
Okay, so water could be a problem, right? If we take away or we don't have enough of some of these things, that's going to be a problem. Water probably not, because water is pretty much 55 molar all the time, right? Think about water, it's so much water, right? I mean, if you run out of water, you probably die before the water concentration changes much. So what else? But the concentration is the right idea.
Not enough bicarbonate.
Not enough bicarbonate, right? If you don't have enough bicarbonate, if I didn't have enough bicarbonate, well, this way would work that way. But so this way, right, I could just keep pulling this out. But if I ran out all my bicarbonate, can I actually shift the reaction this way? I can't consume this H plus. I can't take this H plus out of solution by making the carbonic acid if there's no more bicarbonate left. Not something that really happens in your body, but this is what happens in buffer systems. And so we can look at this with, so what you want for a buffer to work, it's going to buffer best at the pKa when you have high concentrations of both the acid and the conjugate base. Okay. I'm going to say when near the pKa of the acid, and when there are relatively high concentrations of the acid and the conjugate base, both of them. Let's take a simpler system. If we look at acetic acid, so acetic acid is a weak acid, and actually I should say our mixtures of, let me modify this up here. Yes, our mixtures of a weak acid and its conjugate base. Because if it's HCl, it's a strong acid, it just all becomes H plus and chloride and it can't do anything, right? There's no real equilibrium there. I mean, there is an equilibrium, it's just all on one side. So if we take acetic acid here, acetic acid can produce acetate, and this is the acid that you'll find in vinegar. The difference between all vinegars is just other ingredients that give a little bit of flavor. But acetic acid has a pK of it's either 4.25 or 4.75. I'm going to call it 4.25, but I might be wrong by half a pH unit. Did I say 2.5? We'll do that. 4.25. All right, that's our pKa there. And so if I was to dump acid into this solution, what would happen? Let's say I'd add HCl. HCl fully dissociates, so I'm just getting H pluses. Right? If I add H plus, I'm going to get more of this, and we're going to shift that way, right? So add HCl, strong acid. What if I add a strong base? So we're going to go ahead and we're going to add sodium hydroxide, right? Sodium hydroxide, the hydroxide is going to come and it's going to grab this, right, and make water. And so we're going to drop that, and it's going to go this way. All right, but do you see that if we look at, you see that if I don't have any acetic acid left over, and all I have is the conjugate base, if I add a bunch of base to it, there's no more acetic acid to dissociate to replace that. Okay, and vice versa. And so I say relatively high concentrations. I don't expect you to know those, but just in case you remember and you're working in a lab or doing research or something, 10 millimolar is about where it doesn't cut it anymore. Okay, so if you have something that's 50 millimolar, probably good. 10 millimolar is really kind of, and below is really iffy when you're talking about a buffer. At least for biological systems. Okay, questions? All right, so what do I mean when I say near the pKa? There's actually, we can think about that. Here we could use our Henderson-Hasselbalch equation to help us out. Our pH is equal to our pKa plus the log of A minus over HA. All right, and I want you to think about something. So when pH equals pKa, right? The concentration of A minus how is it related to my HA?
They're equal.
They're equal, right? Great. So we have equal amounts of each. That's pretty good for a buffer, right? If we want this reaction to be able to go each way, we'd like to have equal amounts of each. What if the pH is 1 greater than the pKa? Okay. Well, you've got a bunch more acid, but what is this ratio going to look like? Let's kind of do that as a calculation. Okay. All right. To make it easy, we'll put numbers in, but do you see that? Well, actually, we'll not really put numbers in. So let's say the pH is 1 away, right? So it's a 7 as opposed to a 6, right? Or it's an 8 as opposed to a 7, okay? Or it's a 105 as opposed to a 104. When I subtract pK from both sides, what do I get on the pH part? 1, right? So in that case, so pH 1 unit higher than pKa. We're going to have 1 is equal to the log of A minus over HA. And do you remember that what we do is we want to get rid of the log, we're going to take it to the 10. Well, what's 10 to the 1? 10. Thank you for not getting your calculators out for that. So what that means when we get rid of this, what we've got is 10 is equal to A minus over HA. So you see we've got a 10-fold ratio between the two. Can you see if you were two units out, you'd have a 100-fold ratio, right? Okay, and and on the other side, if this was -1, it would be a 1 over 10. If it was -2, so if you're 2 units away, you've got a 100-fold difference between your acid and your conjugate base. If you're 1 unit away, you've got a 10-fold difference. All right, so generally, when we say near the pKa, we mean about 1 pH unit. All right, so when we say pH is near the pKa, we generally mean about 1 pH unit. And so when you get beyond that, things start getting iffy, right? You might start getting worried about your buffer being good in maintaining the pH. Anybody ever for like research or prepping for a lab have to actually actually have to take the acid and drip it in, or take the base and drip it in and get the right pH? You guys haven't lived. If you end up doing that, what you'll see is you'll be trying to adjust the pH, right? And as you get closer and closer to the pKa, you get more of the equal amount of the conjugate acid in the conjugate base, right? And the pH slows down. So you're like a long ways away, and the pH is moving fast as you add acid. Let's say it's coming down. And then you get close to the pK, and it slows way down because it's really good at buffering there. Maybe. But if you ever do lab prep or anything, or ever do research, you're going to do that. So you'll get to experience that. All right. So let's go on. Anything about buffers before I move on? All right. Then I want to remind you where we were with DNA. So we had finished talking, or not DNA, but nucleic acids. So nucleic acids form polymers. Okay, and I'm going to ask you to draw the structure of a nucleic acid showing all atoms, or I'm going to ask you to draw a base pair, and I'll talk about base pairs later, okay? But I will absolutely ask you to draw the structure of a nucleic acid sequence and I'll say RNA or DNA or whatever, or the structure of a base pair on the next quiz and on the next test. Okay? I always do it. I've never not done it. I never do both. I ask for one. Yes. And
So
go ahead.
the practice that we did the other day where we were kind of given the structures and then we just had to reconstruct the sequence.
Yeah, you're going to have to draw good from whole cloth. All atoms,
Okay.
no help. Okay? So you need to know them. And I was having people draw it with the structures up because you hadn't seen them before, right? But I wanted people to get practice with, hey, this is how the connection is. I had a number of people who lost this hydrogen here. I had one person who used to attach this carbon to there instead of there. A lot of common errors I see every year, and I saw them when people were practicing, when they could see it. So I wanted you guys to kind of get an idea of, okay, this is at least how difficult it is going to be for you, or what it feels like. All right, so you want to practice that. I don't really post practice problems for that. You can look at a structure and write it, or if you want, you're welcome to draw some structure and say, I think this is the sequence of whatever it is. Take a picture and email it to me, and I'll look at it. Okay, I'm happy to do that. I'm sorry, Jacob. What? Did I say hydrogen? I meant carbon. This carbon. They go straight to there. There are other mistakes that happen as well. Okay. All right, so you need to know that. So I also was yesterday saying that RNA was less stable than DNA, and it was because of that hydroxyl. That's why I consider the hydroxyl to be the really big difference between DNA and RNA. It causes big structural differences. It also makes it lots less stable. The T and the U isn't a big deal. Okay. So RNA is much less stable than DNA. Because the 2' hydroxyl can act as an internal nucleophile. What's a nucleophile?
Really likes nucleus.
Something that likes a nucleus or something positively charged, right? And so what we've got here is this is the mechanism. All right, I apologize for biochemistry here. Whenever you get these textbooks, they're like sloppy with their electrons. All right, but we've got a pair of electrons here on this hydroxyl. It's going to come in and it's going to be just some base, any kind of base out in solution. It comes in, you can get that, you pull out this, and now we've got a hydroxyl, right? We've got a negative charge on an oxygen right here. Here they're showing the direct attack. I'm not sure whether you get it there and then it attacks, but you get this attack on the phosphorus, and then you're going to displace the rest of the strand as a leaving group. Okay. Does that make sense? What would happen and why? I'd like you need to be able to draw that mechanism. If I ask you to draw this, I'll like give you this without the arrows, and I'll say, draw the arrows and show me the product. And I sometimes do. Yes?
Or is the OH just coming from like water or something added to it, or?
Oh, it could be just water.
Okay.
You hang out in water long enough, you're going to see an OH like this. Okay. Why might this phosphate right here be a little bit shielded from an OH? What's that? Sterics? Maybe sterics. What else? So electrons. It's about electrons, surrounded by electrons. We got a negative charge here, right? So there's a negative charge close. So if a hydroxyl kind of comes close, it's going to get pushed away, right? Can this hydroxyl, once it's deprotonated, get away? No, because it's attached. So you got one, it's held right there. So eventually it's going to happen, right? So DNA can last and be stable millions of years, right? You've probably seen one of the Jurassic Parks, right? Do they actually take the DNA out of amber after the first one? I stopped watching them. But you know, you can get DNA samples that are super old. You can't get RNA samples that are super old because of this. And that fits the nature of what RNA is. We talked about this before. We said we've got DNA, right? We go DNA, RNA, protein. All right, I'm going to win. DNA, RNA, protein. And your DNA is your blueprint. It's your master copy. You don't want it to change. Right? This is cancer is a disease that of the DNA. Your DNA gets changed somewhere in one of your cells. And then the cells decide they're going to be just, your cells stop being altruistic and they get out for themselves and just make copies of themselves, essentially, right? Your cells, when something's bad going on, they're supposed to take a hit for the team. And cancer's when they decide to stop taking a hit for the team. But so you get that. Your DNA, so that's for you, and also for passing on your DNA to your offspring. That's supposed to be unchangeable. It is not, you don't get progress by mutations for the most part. You get progress by taking the genes we have and kind of swapping them around in controlled ways. Okay? Not you get a cosmic ray that comes in and damages your DNA. When that happens, your DNA is supposed to get fixed. And there are repair mechanisms for that. RNA, though, is the kind of disposable photocopy that goes out to the cells to make the proteins. And so that it's okay that degrades. Right? And we'll see later that, we'll see second semester, that, you know, when you make copies of DNA, there are all these mechanisms to make sure that a really good copy gets made, that's perfect. Okay? And when you make copies of RNA, who cares? It's not horribly bad, but it's when you make copies of RNA, it doesn't matter so much, and there aren't huge systems built up to make sure that everything gets done well. Okay, questions? All right, so here we are, we're at base pairs. You need to be able to draw a picture of the base pairs, maybe. Or you need to be able to draw a picture of you need to be able to draw a structure of the base pairs, or you need to draw a structure of the strands, but not both, and certainly not both at the same time. Okay, sometimes people draw both at the same time for me. And that's lovely, but that's not what I'm asking for. We don't need that much art skills. Okay, so DNA forms these double stranded polymers, right? What I want you to notice is that the polymer here is anti-parallel. All right, which means that it's going different directions, right? This goes 5' to 3' in this direction. This strand goes 5' to 3' in that direction. This is just a cartoon over here, right, of what we're looking at. But then here we've got the structures with all atoms. These C1s are these connections right here. So when you think about drawing these strands, this is going to the backbone. All right, 2 backbone. That's where you connect the backbone there. And then you can see that we have our thymine and our adenine can come together and they can hydrogen bond. They can make 2 hydrogen bonds to stick together. Okay? Our cytosine and our guanine can make 3 hydrogen bonds to stick together. And that's how these match up, and you don't end up with mismatches, mostly. I'll show you in a second what else goes into this. Okay? Because there are other ways you could arrange these to make hydrogen bonds. Questions on this so far? So you will need to be able to draw, a base pair, I don't know which one I'll ask you, showing all atoms and showing the hydrogen bonds. And you need to indicate the major groove. All right, so the major grooves, that we have 2 grooves here, you'll see them in the 3-dimensional structure as well. The major groove is this. And the minor groove is this. And when I say this, I mean the space. So these are base pairs. The base pairs stack. We'll look at a 3-dimensional structure in a second, right? They stack, and so when they're stacked, this side is the major groove. You see how there's more space to bind things before you run into backbone, as opposed to the minor groove where there's less space. It's narrower. It turns out to be deeper in the 3-dimensional structure as well. The smaller one is the minor groove.
Yeah, like the top and bottom.
I could draw it in any orientation I want. Or you could, actually, if you're drawing it. But, so what does that look like in 3 dimensions? It looks like this. So if we have our DNA and it's in 3 dimensions, we have this structure. Okay, and so in cartoon form, it looks like this, but you can see it's the same. And the major groove, you see this big wide open space? So if you're designing drugs that you want to bind to DNA, they will bind to either the major groove or the minor groove, but the shape of these grooves is different. I've got more space here and less space here. And the functional groups are different, right? In different, at least on different sides. So if we look at the minor groove of these, right, we've got a carbonyl here. We don't really have, well, here we actually do have an amine. So if we're wanting to get a guanine cytosine base pair and we want to target that, we maybe look for something that would bind or hydrogen bond with this amine in the minor groove. So does that make sense? You'd be looking for specific functional groups in different orientations if you're doing drug design. Questions on this, what you need to know here. Okay? So this class has a bunch of memorization in it. They're just some things that you need to really be familiar with. So we've got this DNA, what does a nucleic acid structure look like overall? What do these base pairs look like? Because there are things like, there are types of damage to happen to these base pairs that are important. There's all sorts of reasons. If you think it's a lot of memorization now, just wait until we get to amino acids. You'll notice with this, so we've got that. I don't care that you know kind of the step size or anything like that, that it's 3.4 angstroms, that it's actually 10 bases per term. But I do know that I do care that you know that it's a double helix, right? Okay. I care that you know that it's anti-parallel, all right, in terms of the way directions of the strands. And I care that you know that it's right-handed, and you can recognize that. This is a right-handed helix. So we're going to learn how to do that. This is a form of chirality. All right, so how do I know that's a right-handed helix? Okay, and in grad school, I worked in a group that did drugs when I did DNA. And so we would look at the cover of every magazine when they put DNA on it, and they'd have it. They take the image and they take the negative and they put it on, and it would be a left-handed helix. So that bothered us, of course. But we do have right and left-handed helices that we see in a number of different contexts. So right-handed helix, the way you do that is you take your hand, okay? You point your thumb either direction, it works, okay? But you take your thumb's pointing the direction of the polymer, and then it's right-handed because as the polymer moves in the direction of my thumb, right? It's going like this. Do you see it's going like this? It's my right hand matches. If I try and do it with my left hand, it's against the way it's going around. Okay? Not everything is right-handed.
Is DNA always right-handed though?
Yes.
Okay.
DNA is always right-handed. But if you want to look at something and recognize whether it was right-handed or not, here's some practice. Okay, so we've got A and B here, and then we've got something over here. This is actually one of those stereo images. If you cross your eyes just a little bit, you'll see it in 3 dimensions. But you don't need to do that. You can just look over on the left-hand side. A is one chirality, right? B is the other chirality. Which is which? Use your hands. A is right-handed and B is left-handed. Okay, great. Oh, I forgot one. Hold on. No, we can't forget it. There. How about that? Right-handed or left-handed?
Yeah.
How many people think it's right-handed? How many people think it's left-handed? Okay, it's right-handed. Okay? Lots of right-handed, huh? Just in nature.
I was just looking at the center of the shell. It keeps turning.
Okay, questions on that? Everybody see how to do that? You want me to post a practice worksheet?
Yes.
Okay, I'll do that.
Will this be on a quiz?
It's always.
Oh, it
yes.
could be on a quiz.
I think you should put it on a quiz.
Yeah, it's not as important as the other things I'm talking about. It might show up. It might not.
You have to ask yourself, do you want practice questions? The answer is yes.
All right, so I guess I just lost my notes altogether. Give me a second. And I'll get our snail in here. All right, so the snail was right-handed. Okay, and we were saying this one was right-handed, and this one was left-handed. Okay, and I'll put up a worksheet. All right, so this is where I talked about how it wasn't just the base pairs. It's not just the number of hydrogen bonds for base pairs that causes you get A pairing with T and G pairing with C, okay, when these things polymerize. Now, how many people haven't heard that before? Okay, you haven't heard of that before? So A pairs with G and C pairs with T, and that's all you get. Let's roll back to that real quick. All right, so you'll notice here in DNA, whenever you have a C, you've got a G across from it, right? There's another C, a G, Okay, C across from G always, A across from T always, but you know the A could be on either strand, but then you have a T on the opposite strand for that. And that's based on this base pairing, but it's also based on the shape of the enzyme pocket. So A pairing with T and G pairing with C is both the hydrogen bonding, or is a combination of the hydrogen bonding and the shape of the base pair in the enzyme pocket. All right, so what enzymes are is, when you think about it, I like to think about enzymes as big old Pac-Man, big little mouths. So you got essentially what you have is you have a giant protein here, and you have a little active site, and the whole point of that protein is to hold functional groups in the right position. It's all structure to hold functional groups in just the right position over here, to make this just the right shape for things. Okay. And so I'm not going to draw the same thing here because I don't have room on this on the screen here, but these are the natural base pairs. Okay. I'll just call them the regular base pairs. These are the base pairs that Watson and they're called Watson and Crick base pairs because Watson and Crick came up with them. And if you look at the pocket shape here, do you see that if I draw the same thing here, I start running into atoms? So I've got a pocket on my enzyme. Here, maybe I don't run into atoms, but I've got extra space. It doesn't fit as well. You know, it depends on where you start here, but same sort of thing here. So this would be the shape of the enzyme pocket. So that's how you get this A pairs only with T and G pairs only with C. Okay. There are a couple other mechanisms downstream of that even, but this gets 99.9999 whatever percent of it.
So are you saying that the major group is the enzyme pocket? Like what part of it?
Yeah, I'm not sure. I'm not sure exactly
Okay.
where the enzyme is doing it. So if yes, if my little picture here is correct,
Okay.
right? But it's going to be more complicated than that because my picture doesn't exactly work if you flip these, right?
Right.
Never had anybody really ask me about that, but I'm giving you it's a three-dimensional enzyme pocket that fits these and makes only these work, but it actually works regardless of whether you have a C here or a G here, or a T there or an A there. It doesn't matter for the pocket. It's only one enzyme.
Okay.
Yes.
So is like the enzyme pocket kind of similar to like how it only fits like certain substrates fit an active site, so
Yes.
certain base pairs fit
Yes.
in the enzyme pocket?
Yes. Only if you put a T in the base in the enzyme pocket, only that A is going to fit there.
Okay.
Okay. Based on the shape and the electronic structure.
Okay.
Okay. You guys got that? All right. Then, when we think about how this reaction, so in terms of DNA being polymerized, this is actually the reaction mechanism, and you're going to need to know it. I'll talk about it in a second here. So this is the reaction of adding a nucleotide to a growing nucleic acid polymer. I'll just say to a nucleic acid polymer. All right, and so what you've got is you come in with an NTP. So this could be an ATP, which you've probably heard of. It could be a TTP, a GTP, a CTP, if we're doing DNA. And in fact, for this picture, we are. It'd be the same if it was RNA, except there'd be a hydroxyl here. And so in that enzyme pocket, what happens is a base comes in. You've got some other base over here guiding which one this is. All right. But the mechanism is that you end up with a nucleophilic attack from this oxygen down to that phosphate, and then we're going to kick that up, and we're going to have this pyrophosphate leave as go out as a leaving group. So I'm actually going to draw the pyrophosphate. Probably not the it'd probably pick up a proton or so. I'm not sure what pyrophosphate looks like in solution. That's a lot of negative charge. Okay? So one reason I'm showing this is, first off, the main reason I'm showing this is we're about to start talking about, we won't have time to really get into it today, but we're going to talk about PCR, right? And PCR involves this reaction. You're going to add, you have this reaction. This reaction is really important, so you need to know this as well. I will give this to you, like I'll give you this, and I won't give you the arrows. You'll have to draw the arrows and show me the products, okay, for this. So, but one thing I want to emphasize about this as well, because we're going to see this reaction over and over again in biochemistry, is that whenever we make a pyrophosphate, what happens is that immediately gets broken down to two phosphates. You don't take the pyrophosphate and then put it back onto ATP to regenerate the ATP. You make phosphates and then you'll make ATP taking ADP and phosphate, if that makes sense, right? Not this one though, because this one's now in the polymer. Okay, this reaction right here, for this reaction we have a free energy change of a standard biochemical free energy change of negative 19 kilojoules per mole. Okay, maybe it's 19.5, but it's not. So what is that? What's the use of that? I make a pyrophosphate, and then as soon as I make the pyrophosphate, it gets broken down in this reaction that has the spontaneous reaction that has a free energy change of negative 19 kilojoules per mole. Yeah.
No, I just have a question. Is the structure written in black, that's supposed to be like three of those, and that's ATP.
This is we have three of these, right?
Yeah.
And then this leaves as a leaving group. This part right, whoops, there.
That's ADP now.
Well, it's not ADP. This right here, if this was an A or whatever, okay, this would be now added on to the growing chain. So now we'd have a DNA that had an A on the end instead of a whatever was on the end. Let's put letters on. So let's say this was a C and this was an A. Now at the beginning here, we have a DNA polymer that ends in C. And then after the reaction, we have a DNA polymer that ends in A because we've added an A to the end. And to the 3' end, because this is a 3' end right here. Always to the 3' end. Does that make sense?
Yes.
And then this part, this is actually, so here what we're doing is, so we have this nucleophilic attack here, and this is a leaving group. Okay? So this is going to help us make this connection by having this group leave, and there's our leaving group now. This is that. Good question, by the way. You're confused about it, so are other people. Yes?
So, can you re-explain what those, you said those don't immediately join back into something? They're not just like floating, or they do?
These are immediately, so we've got the pyrophosphate right here is immediately broken down into two phosphates. Whoops, that's why it's confusing.
Yeah, that looks like the same one.
It is the same one. I said two phosphates and I drew another pyrophosphate. There, two phosphates.
Cool.
Okay, now does it make sense?
Yes.
immediately broken down into 2 phosphates, and that reaction is very spontaneous. How is that useful? Okay, we're going to push the reaction this way. Why? What? Le Chatelier's principle. So this becomes, you keep this very low, right? And so you drive the reaction this way by keeping the concentration of one of the products super low, right? So it helps drive the reaction. There's another way we can explain it in our last, we don't have a minute, so I'll explain it, all right? The other way to explain it, this is a coupled reaction, right? This reaction is coupled to this, so I just added 19 kilojoules of spontaneity into the reaction going that way. So that's another way you can explain the same thing. Okay? You guys have a great weekend. We will talk about PCR when we get back.